So it's really a pleasure to introduce Bill Snell. I guess I've known Bill for a very long time because it turns out that people who work on obscure organisms, that is non-mammalian organisms in the cell biology field, tend to sort of hang out together. And so we've ended up at a lot of the same meetings and talking about a lot of the same things. So one of the reasons it's a pleasure, I gotta say that Bill is really a giant in this field. He has dominated, in fact, I would say, single-handedly defined many elements of cell biology of Chlamydomonas mating ever since he was a grad student. So he was a grad student with Joel Rosenbaum at Yale, where he started out doing work on assembling tubulin onto flagellar basal bodies, but then quickly ended up really kind of defining the whole adhesion system in mating of Chlamydomonas, and had two huge papers in JCB. Well, one of them is 21 pages long, defining single author papers as a grad student, defining the way that they adhere to initiate the mating reaction. He then did his postdoctoral work at Hopkins with Saul Roseman, studying the kinetics of the mating process, and then rapidly went on to, so I want to make sure I get this right, University of Texas Health Science Center
Southwestern Medical School. We got to get the Southwestern Medical School.
I thought that was in there somewhere.
Yep.
Anyway, and he's been there working on Chlamydomonas mating for a very long time, and then moved up to join us in the Maryland area, where he's now at the University of Maryland. So he's had an incredibly long, storied career using Chlamydomonas mating to identify deeply conserved cell biological systems that you can access easily using the process of cell fusion. So this is very near and dear to my heart as well, of course. One of the things that he has discovered is HAP2.
Consumers request a louder volume.
HAP2, which is a highly conserved plasma membrane fusion protein. GEX2, a nuclear envelope membrane fusion protein. Both of them deeply conserved, highly conserved. And that, I think, is a perfect introduction to his talk, which is entitled Using a Unicellular Organism's Flirtation with Multicellularity to Uncover Deeply, Broadly Conserved Mechanisms of Fertilization. Bill.
Thank you. Thank you for a very nice, generous introduction. Can you hear me okay? I guess I'm on. Well, I have been studying Chlamydomonas for a long time, and actually I became interested in using unicellular organisms to study cell-cell interactions as an undergraduate studying tetrahymena mating. And when I moved to start my graduate school work at Yale, I realized that 7 mating types was pretty complicated. 2 was good, and Chlamydomonas had 2. And so I've been working on Chlamydomonas reinhardtii since then. And what I'm going to end up telling you is that with clammy seems to have become a great unifier in terms of reproduction and understanding reproduction. And one of the things is that its position, I guess, in evolution, it can actually look back, maybe if you have to look back to see viruses, and it looks forward, and we can see vertebrate biology of reproduction here. And so during my talk, there isn't a pointer around. There was a pointer here, but it seems to be dead. And I could use my screen, I guess, but I'll do it this way. So I've got 3 sets of, there we go. I just didn't know how to do it.
Button right in the middle.
There are 3 sets of molecules up here that I'm going to talk about during the course of my presentation. And in the center of each of these is a Chlamydomonas molecule. This is a Chlamydomonas actual X-ray structure. This is an actual X-ray structure. These other molecules or the other images are predicted structures based on the distance-based method of a software system called Raptor X. It's probably not quite as good as the newest AlphaFold, but I'm pretty sure it's telling us what's going on here. And it turns out this is a viral protein that I'll be talking about. This is an Arabidopsis protein. This is a Chlamydomonas fusion protein. Over here, we have a protein that's near and dear to Mark Rose's heart, CAR5, which is a nuclear envelope, a nuclear fusion protein. This is Chlamydomonas protein, and this is brambleberry protein of zebrafish. And then over here, we have a predicted structure. These are predicted structures. A predicted structure of Chlamydomonas adhesion protein, fuse1. Over here, we have an Arabidopsis protein, sperm adhesion protein called gex2. And over here, we have a bacterial protein that's responsible for binding bacteria to our cells. And so again, as I was saying, we can look to viruses. We can look both at viruses with Chlammy, and we can look at vertebrates with Chlammy. And so it's really been a neat thing. And all these are reproduction proteins. And what I've realized is that being in the position of Chlammy, Chlammy let us see that the protein that we were studying actually had similarities to these others that other people hadn't been able to notice before. And so that's what I mean by a unifier. And I'll talk a little bit more about that as we go along. So this is what the organism looks like when it's mating. It really is almost all of its life unicellular. But when we mix in the haploid, when we mix plus and minus gametes together, then they undergo this flirtation with multicellularity. They're not a unicellular organism anymore. They adhere to each other by their cilia. They end up getting real close to each other, plus and minus gametes, and then they fuse to form a single-celled quadriflagellated cell. So it looks like this. The two cells come together, they stick, they get their mating structures, which I'll show you in a second, close to each other, and then they fuse to form a single cell with 4 flagella. That cell brings its cilia in, or flagella in, makes a thick cell wall, and that thing can withstand extremes of drying and temperature and other kinds of environmental stresses until conditions get good again, when it can grow vegetatively. Whoops. So what I'm going to talk about today is how we've used Chlammy to learn about sexual reproduction. And this is just a reminder about the importance of sexual reproduction in humans. This is actually a picture of some tossling corn at our farm in Illinois. And soon after we discovered HAP2 and what it does in Chlammy, and I realized it was present in corn, in pollen of corn, when I go back to our farm in Illinois, when it's corn tossling time, I feel like I'm surrounded by my molecule, and it's sort of a mystical experience. Even it really was. I got goosebumps when we first did that. And then, of course, fertilization in eukaryotes is important. And one of the things I'm going to talk about is the importance of sexual reproduction in Plasmodium, the malaria organism, that occurs in the gut of the mosquito. Really exciting, nice place to be if you want to do sex, I suppose. But and it's critical for transmission of malaria. And so that's one of the things I'll talk about. And then, of course, this is what when we think about fertilization, often we think about an image like this. This is an image from Paul Wasserman of sperm interacting with an egg. And what I hope to communicate today is that we now know more about what goes on during fertilization and sexual reproduction in Chlamydomonas than we do in any other system. Let's go back here. We know what the sperm adhesion protein is probably, and we know what the egg adhesion protein is, but we don't know yet what the fusogen is in our sperm egg system, which is sort of amazing when you think about it. We don't know what the molecule is that brings our sperm and egg together. So almost all organisms use sort of a basic set of steps for fertilization. We start out with haploid gametes. They adhere to each other, they interact with each other and recognize each other. Usually, not usually, but in sperm, the plasma membrane interacts with the outer vestments of the egg and activates the sperm. In Arabidopsis, it's maybe the other way around. But anyway, they get rid of the things that are preventing their membranes from getting together by this activation process. And then the plasma membranes undergo an adhesion event. And then there's bilayer merger. Then there's pronuclear fusion with a little bit of an asterisk. It turns out our cells, we don't do that. Our pronuclear membranes break down before the first mitosis. So there's never any nuclear fusion. And I was perplexed by the fact that we didn't find GEX1 in vertebrates until, I'm sorry, in mammals until we realized that. And then we have the zygote that's then going to go on to do whatever it's going to do after that. And I already mentioned that the gamete membrane fusion reaction in mammals, we still don't know what the fusion protein is. We do know the receptor pair on Zoom1 sperm and Juno on the oocytes. And it turns out for all the other organisms, we don't know them yet. Mark has been, and many other people have been working at it in yeast for a long time. C. elegans, we don't know how it works yet. Fish, we don't know how fusion, gamete adhesion and fusion works. It turns out flies have HAP2, which I'll tell you about later. And their sperm, there's a master's thesis that's not been published besides being a master's dissertation from Barbara Wakimoto's lab, who shows that HAP2 is essential for fertilization in flies. And as I mentioned, what we know is that this initial recognition event is species specific. And then there's this membrane adhesion event, and that's species specific and conserved. And I'll explain what I mean in a little bit. How can it be species specific and conserved? And then bilayer merger is broadly conserved, except we don't use HAP2 in chordates. And then this nuclear envelope fusion is broadly conserved. And so I'll come back to those things. So we're going to start by talking about CAR5 GEX1 brambleberry. But first I want to let you know about that one of the things that we've been found so useful is a transcriptome. We've identified a set of genes in Chlamydomonas that are gamete specific and that are even specific not only to the gametic phase of the cell's life cycle, but also even we know which genes are upregulated in plus gametes and which genes are upregulated in minus gametes. And I realized that I did not show my life cycle diagram. But anyway, it turns out that we grow Chlamydomonas cells vegetatively, asexually in the laboratory. And there are minus gametes and there are plus gametes. And the mating type is determined by what's called the mating type locus. And if the cells have the plus mating type locus, they're plus, minus mating type locus, they're minus. And if we put them in nitrogen deficient medium, they differentiate into gametes. And then when we mix plus and minus gametes together, they stick to each other by their cilia to form the zygotes that I showed you before in that one of the early light micrographs. So we looked at we got messenger RNA from minus gametes, from plus gametes, from vegetative cells growing asynchronously and synchronously. We got them from gametes that have been activated to undergo the events that they need to undergo to to be able to fuse. Well, we could do it in cells of a single mating type by activating them with cyclic AMP, which is normally produced when they do this. And also when they're getting ready to mate, they have to get rid of their walls. And when they get rid of their walls, they lose their walls, they upregulate wall genes. And we wanted to be able to identify those too. And so we have a series of vegetative cells, plus and minus gametes, activated gametes, and then wall synthesizing gametes. And that has been hugely valuable for us. And when we first did this and did a cluster analysis and identified some that were this series over here are minus gamete specific, one of the ones we saw was GEX1. And GEX1 was identified originally in Sheila McCormick's lab in a rabbit. Actually, it was first identified in maize. She used methods to see which genes in maize were upregulated or gamete specific. And she found a series of them. And then one of those, one was called GEX1 and one was called GEX2, which you already heard me mention today. And so she had this GEX1 and we noticed it was in clammy. We thought it was in clammy anyway. And so we screened, I don't know, 40 or 50,000 insertional mutants for strains that were blocked that had a defect in GEX1. It turns out that we did not see it. It was expressed in minus gametes, but it also has to be present. It's also used in plus gametes. So what we found is if we use the knockout and we mix both plus and minus gametes that had the knockout of GEX1, we got no meiotic progeny. Mix the cells together, they fused just fine, made zygotes. Zygotes pull their cilia in, made a wall. They would, when we added light to them, they would hatch, but nobody grew any progeny. All the progeny failed to survive. And interestingly, GEX1 was also present in Plasmodium, the malaria organism. And we worked with Oliver Bilker, who was a fellow actually who originally convinced us and worked with us to do this transcriptome. And they knocked GEX1 out of Plasmodium burgii, had the same phenotype, sort of mind-boggling, in that they underwent good zygote formation, but the zygotes failed to be able to produce progeny that could get out of the wall of the gut of the mosquito up to the salivary gland, so it could inject those cells into a human, or in this case into a mouse. Same phenotype. And in clammy, the protein we rescued with a mutant with the tag form, and it was present in the nuclear envelope. And same thing in Plasmodium. It was present in the nuclear envelope, and then after fusion, you could still see it. And in clammy, when we did electron microscopy in a wild type plus and minus mix, this is the whole cell. This is a zygote. Its nuclei at 7 hours have fused. You can still see the two large pronuclei here. But in the GEX1 mutants, the nuclei have not fused. It turns out a lot of them do end up going ahead to fuse, but still the progeny are blocked. And so this is what we saw a lot of. The cells, the nuclei just did not fuse. I sort of got, because we find these oddball proteins in clammy, I spent a lot of time blasting. And what I realized when I started blasting is that not only was clammy similar to yeast CAR5 and Arabidopsis and Plasmodium, but all these organisms had a GEX1. And so the people that had, Abrams et al. Had discovered brambleberry in zebrafish and knew that it was responsible or participated in nuclear envelope fusion. And they saw that it had some similarities to CAR5, but they didn't realize the whole protein was similar to CAR5, and they didn't connect with GEX1. And so what we came away from with this was a neat example of, again, how clammy could help us look in many different ways and discover that this protein, which when we did homology modeling, when we did distance-based modeling with Raptor X, they all look like this. So here's clammy, here's CAR5, and here's brambleberry. Very similar structure, transmembrane domains at one end, some of this stuff at the other end. You can sort of just guess it's going to be bridging something. So it should be neat to figure out what the structure, how the structure is connected with its function. Okay, so that's CAR5. Now I'm going to talk about our work on a second protein that we discovered that I mentioned earlier, which is this GEX2 protein that Sheila McCormick came up with in that screen for maize that then she took to Arabidopsis. And Fuse1, the protein that we've known for a long time on Chlamydomonas, is essential for mating structure adhesion. Yep.
Just one quick question.
Sure.
You mentioned that the GEX1 is only expressed in the minus gamete.
Correct.
Can you just, and so there is no, so there clearly would not be any consequence in the Chlamydomonas knockouts on the plus gamete side. And so maybe you're getting this, but there must be a recipient, right? There must be something on the plus side that GEX1 is looking for, and maybe a binding partner of sorts. But I actually just wanted to ask if you could give us a definition of the why the designation of plus and minus, what the meaning of that is. Is it just?
I guess the convenient way to distinguish one type from another type in turn, it's an operational definition. Pluses can stick to pluses and minus can't stick to minuses, but pluses can stick to minuses. And the mating type locus has about 7 different genes, and they're scrambled in their alignment, so they don't undergo crossovers there. So it stays as a little unit. And it turns out the minus mating type locus has a transcription factor called MID, that if it's missing, minus gametes look like plus gametes, except minus gamete mating type locus doesn't contain fuse1, which I'm about to talk about. So that's how it came to be.
There's transcriptional control.
That's right. That causes one set of genes to be turned off and another set of genes to be turned on. All the other proteins, like fuse1 gene, the fuse1 gene is present in both mating types. The ciliary adhesion molecules are present in both mating types. The genes are, but they're only turned on in one or the other. And now about the fact that GEX1 seemed to be expressed only in minus gametes as a transcription factor. We were surprised when we did the knockout that when we did it, I think originally in minus gametes, although I don't remember which one, and they were perfectly fine in fusing. So that was really with the wild type of the other cell type. But once we had the tag, and we could look at cells soon after they fused, or when they first fused, you could see one nucleus that was labeled and the other nucleus was not. Very quickly, both nuclei got labeled, even before fusion. So we're pretty sure that once they're in the common cytoplasm, the gene is being expressed, and it can end up in both, because it looks like then, if we knock it, only when we knock it out of both, do we get a blockage to fusion.
And I would just say, it's exactly the same situation in yeast. Knock out CAR5, one parent doesn't.
So there's a whole neat biology there that Mark started a long time ago that's just going to be neat to figure out, and it's a really tough thing to get at.
And then to follow up with that, do you get are clammy-like Saccharomyces in the fact that they're pretty much exactly the same except their mating type? Because I know like the difference between the two cells in Saccharomyces is like that's it. It's just the mating type. So
When they're vegetative cells, there's just ain't no differences. When they undergo gametogenesis to turn into gametes, they do express different sets of genes then. If you look at them before you've mixed them, they're swimming around, they look the same, motility is the same, the cilia are the same. Ultrastructurally though, they have these, well, you can see that here. This is a plus gamete, and it's been activated. Flagella are out here, cilia are out here. This guy's cilia are out there. It has this little region before activation. This thing is not elongated like that. And so there's a little dark area right here that sort of sticks up a little bit. You can see that. And the minus gametes have a similar place where there's this little donut. If you could look in there, it'd be like that. And they look a little different. And here they're a little bit more different. Those are the basic morphological differences, though, really right there. Okay. So when we mix them together, they interact with their cilia, they send signals to the cell body, and that's one of the things that we actually, another part of the lab spends a lot of time trying to understand how ciliary adhesion activates a process in the cell, in the cytoplasm. But anyway, they then adhere to each other by the tips of their mating structures. And we know this is a plus because it's got actin in it. This one does not. That's the minus. It's a strong adhesive interaction. And then right after that, the tips of those two things fuse and they come together. And this is caught when they're just about ready to undergo this kind of a reaction. So then probably this actin helps maybe pull this together, but nobody's really looked closely at that. Although, yeah, they haven't.
So are those cilia cylinders with membrane on the outside, actin and motors on the inside?
This is a cross section of a cilium here.
Okay.
It's not a very good one. You can't see the 9+2. Is that your question?
No, my question was in terms of the large scale structure of the cilia. I look at the other images have tubes.
Back.
One on the right, for example, it's a plus gamete cylinder.
Yeah, this is the this
No. I think it's X plus and minus gamete.
right here.
That is are those actin-filled cylinders are surrounded by one bilayer, two bilayers?
One. So it's a microvillus.
Right.
It's a microvillus.
There are motors in there pushing them around.
They don't. We don't know that they move much at all. I don't know if there's any myosin there. And this thing forms within 30 seconds, probably. We can see when it's starting to form a bunch of vesicles going out there, presumably to add the membrane. You add cytochalasin and it blocks it. There's an actin mutant, makes floppy fertilization tubules, doesn't fuse very well at all. So
But when you do this kind of EM, do you have to remove any cell wall or extracellular?
these guys have been mixed together. They shed their walls on their own.
So they're totally comfortable in the osmosis.
Yeah, they are. Yep, we're lucky. They don't have to stay dressed while they're doing sex, like yeast do. Sorry.
Mark, just to be really mundane, is it possible to tilt the camera in the room up a bit so that the titles of slides are being projected for somebody? Beautiful. Thanks, Tom.
Thank you. So now about this step of membrane adhesion. After we've got the gametes together, they're activated, they got their they form their mating structures. Now the mating structures need to interact, as you see here. And again, we can tell which is the plus because it's got the actin inside of it, and which is the minus. And many years ago, Ursula Goodenough, another Chlamydomonas researcher who has written some widely used genetics textbooks, got a mutant called FUSE1 that fails to undergo fertilization, fails to fuse. They thought it probably had something to do with mating structure adhesion. Single pass membrane, transmembrane protein contains, we found that it contained bacterial-like IG domains, species limited, but that's what I'll talk about. And those FUSE1 mutant gametes are incapable of membrane adhesion or fusion. And the FUSE1HA gene rescues fusion, we showed. And we also then went on to show that the protein is present. We can activate gametes of a single mating type, as I mentioned with cyclic AMP. They make their long fertilization tubules and FUSE1 antibodies. Those are antibodies. Bind to the fertilization tubules. You can see stained with actin. That's obviously they're co-localized. And the FUSE1 mutants are missing that protein. And this was a docking assay. We set up a way that we could have ciliary adhesion defective mutants that still had wild type FUSE1, and we could set up a docking assay, show that they could adhere if they had FUSE1, but if they didn't have FUSE1, they couldn't adhere. And of course, they could not fuse. So that was evidence that FUSE1 is an adhesion protein. We did that a long time ago. Just recently in my lab, we've been working on a protein that I'll come to in a minute that binds to FUSE1. And as part of that, my postdoctoral fellow in my lab, Jennifer Pinello, was looking at that. And we knew that clammy had these IG-like domains that occupied about 2/3 of the protein, we thought. And then as we started doing more blasting, we saw that maybe there were some more IG-like domains. And then Jen noticed that it was very similar to this GEX2 protein that other folks had shown was the sperm adhesion protein in Arabidopsis and present on many other plants. So once we started doing more hard, maybe not so stringent blasting using delta blast, it turns out that finding with FUSE1 as a query, we could find other algae that had it. But then down here, we started seeing the first hits in plants outside of algae was GEX2. And if you went down a little bit, we found Arabidopsis. If we use Arabidopsis GEX2 to search the Chlamydomonas database, same thing, FUSE1 comes up first. So and then I showed you this before, but when we ask RaptorX to model them for us, this is ClamyFuse1. It's got predicted 6 or 7 IG-like domains. GEX2 has about 6 or 7 IG-like domains also. Similar kind of predicted structure. Of course, we don't have the structure, and this could be wrong, but I think it's pretty amazing that these two things that were so separate, and the sequence identity of these is like around 18% or something. I mean, it's just, but you can see it. And then this is a bacterial intimin, as I mentioned, or invasion that bacteria use. So that what we've arrived at, let's see. What we think is going on here, if you look at, so this is really an immunoglobulin-like molecule. And our antibody system uses this basic structure to tweak a little bit to be able to bind all kinds of other proteins. It's really a malleable protein. As you know, I don't know how many genes in us are devoted to making IG, like proteins with IG-like domains. I think they're probably evolutionarily related. It's hard to tell yet, and I don't know how we'll end up actually knowing that, but it sort of makes neat sense that a structure that we use a tweak to be able to bind a bunch of different things, evolution seems to have used to be able to bind new adhesion molecules during speciation, even maybe. I mean, I know that's a big speculation, but it sort of makes sense. You have this basic structure, you make this one a little bit different or this one a little bit different, it can bind to another kind of a molecule. And I'll come to the molecules that these things interact with, at least one in just a few minutes.
Is this in the plus gametes or the minus?
This one is in plus gametes,
It's the same place where the ant.
which is the same place where the actin is.
It's also complexes with actin and muscle. I mean,
So this is on the outside. These are only the ectodomains.
So they don't interact with the actin at all.
I can't say that. There's a very short cytoplasmic domain. I think it's only like 10 amino acids or something. But it does. And we haven't looked inside yet. This is only on the outside.
And is FUS1 similarly conserved or X1 in clammy? About 18%.
I think it's very low. Yeah, when we first started talking with people, I said, "Oh, come on, you're making that up. Come on." But I think it's, actually, I started doing all this blasting when we discovered HAP2, and HAP2 also has about the same level of sequence similarity to these other ones. But then I began to recognize, wow, these cysteines are just placed like this, and there's this double cysteine right here, and there's another cysteine there. And so I sort of got to trust that what I was seeing that way was probably correct. And I think it turns out that was the case.
So you said that these are adhesions. So are they adhering on the sides of those beta sheets or on the ends of the beta sheets?
Yes. We don't know. We literally just came across this similarity several months ago now. And my fate might have been decided this morning by the NIH study section that's reviewing this grant. I don't know. If it doesn't work, I'm going to resubmit. But it turns out that we developed this relationship with a group at the Pasteur Institute because of HAP2, the fusion protein that's present in malaria also. fellow's name is Felix Ray. He's an outstanding, wonderful human being and an outstanding structural biologist. He got some of the first structures of the alpha viruses that cause that all the viruses that cause yellow fever, dengue, Zika, all those are class 2 viral fusion proteins. And he's become interested in this whole system. And as I'm going to tell you in a few minutes, the protein that we've identified the protein that the Chlamyfuse 1 interacts with. It looks like it's species specific, but it's interacting with the fusion protein, which I better get on. So now to move on to HAP2. We did another screen for mutants that were, again, around 50,000 picks we had to do, screens, and we found one 63B10 that could undergo ciliary adhesion just fine, but it was blocked in fusion. These are wild type. This is a wild type zygote. These are the mutant that was blocked, which turns out to be the HAP2 protein missing. And HAP2 is a single pass transmembrane protein also. It was first identified in a screen for male sterols by Mark Johnson in Arabidopsis. Several other people, Mori, this Japanese group led by Mori, and Lilly found it also. They gave it a different name, generative self-specific. And while we were working on our stuff, these guys also came out with it. And then Mark Johnson, as I mentioned. And so it was a hot thing. It turns out Mark didn't know what it did exactly. He knew it was important for fertilization. And Maury's group didn't know what it did either. They thought it was an adhesion protein, but it's so hard in plants you can't really work with isolated gametes. And so with Chlammy, though, as you've seen, we can distinguish mating structure adhesion from fusion, and we could show that our mutants, the images I showed you before, are actually all with a HAP2 mutant, because this happens so quickly in wild type you can't stop it. So adhesion was blocked there. This kind of image with this mutant really. It was a good lesson for me, the power of electron microscopy. Sort of says it all right there. This mutant is blocked at the adhesion step. It's located on the minus mating, it's only in minus gametes. Only in minus gametes. Present in both as a gene, but only in minus. No function in pluses. Located at the minus mating structure. And then once I started my blasting again, saw that it's throughout biology, all these different organisms. I guess I do have on honeybee, but also I mentioned when it's knocked out of Drosophila, fertilization fails too. It's in tetrahymen, essentially, it's what lets tetrahymen mate, fuse. And this is work that we were fortunate to work with Nick Grishin at UT Southwestern and Jimin Pei and Rob Steele, also another place, who's a choanoflagellate guy who helped us. It's also present in choanoflagellates, present in all these nasty organisms. So we then wanted to see if we convinced. I called around the world to see if I could find somebody that would knock it out of Plasmodium, because I saw that it was in Plasmodium. And Bob Sinden and his group at Imperial College in London, he was sort of about my age, maybe a little bit older than I am. And he sort of had done a lot of the biology about sexual reproduction in Plasmodium. And this is one of his images showing a Plasmodium female gamete interacting and about to fuse with a male gamete. And it turns out, as I mentioned, this happens in the blood meal. So when mosquito bites an infected person, there are so-called gametocytes that some of which are male and some of which are female, can produce male gametes in about 10 minutes, where they just squirt out these little sperm. Sperm binds to the egg, fuses with it, makes a zygote. The zygote crawls out into the gut wall, makes an oocyst, which eventually breaks up and yields into the saliva the organisms that can be passed on. And so this is a wall of a gut of a mosquito that was infected with wild type Plasmodium cells You can see all the oocytes there. And here are some of the ookinetes, they're called, the zygotes. In a hap2 mutant, no oocysts, no zygotes. So it was really pretty neat. So that's where things stood. We were excited about that. And then we started wondering, now what in the world is the function of hap2? What's it doing? We know it's essential for after the adhesion step. And again, Jimin Pei and Nick Grisham, if you do much blasting, you'll probably come across a Grisham protocol or a Grisham something or other and blast. That's this Nick Grisham. We were so lucky to work with him and Jimin Pei. And Jimin Pei was looking at hap2 and said, you know, using HH Petty, this was, I don't know how many years ago now. He said, I think it looks like a dengue fusion protein. And when he first showed me this, I couldn't even interpret what this was. I just sort of, my brain just couldn't get around what are all these things. And then he said, and there's a fusion loop at the tip. And he said, I think hap2 has some of this kind of structure. There's an IG-like domain right in here. And it turns out the way this thing works, and I'll show you a diagram in a second, this is in after fusion, but before fusion, this thing is down here farther. So it's sort of like this. And this is in the viral membrane. And this is just, actually, this is laying down a little bit like this. And when a virus infects us, like dengue, it undergoes endocytosis. The low pH of the endosome makes it do this. And this little thing out here has these fusion loops, which are hydrophobic residues, that inserts into the membrane of the phagosome inside of us. And then, so now you got it like this. And then they're so-called extended intermediates. And then three of them, these molecules get together and they do this simultaneously to form a trimer, the so-called post-fusion trimer. And so this is one of the molecules in a dengue post-fusion trimer where this is folded up. And so what I just told you is, these are with class 2 fusion proteins anyway. On the viral membrane, they're usually protected by some other protein that in the acidic environment, they stick up, insert into the membrane of the phagosome, begin this bend, and then form this fusion pore. So that's the basic. So they're basically they've got the energy built into them to go like this. You just have to release it. You have to trigger, and viruses, it's triggered by low pH. So before we knew its structure, though, based on what Jimin Pei and Nick Grisham had said, you know, we think this region right in here is probably the one that corresponds to the fusion loop. This is the double CXC that I talked about that I got to recognize. You can see all these cysteines that just sort of, I got to know. So we made antibodies against this, the clammy, and it blocked fusion. Pre-treat minus gametes with it, blocks. Pre-treat plus, no effect. So we were encouraged that maybe they were right about that prediction. So what we went ahead and did then was make antibodies against the falciparum, human form of causes human form of malaria, and the Berghii, the mouse form. And we made antibodies against regions in the fusion loop. And used the membrane feeding assay to test whether or not those antibodies would block fusion. The way this works is you get some blood from an infected mouse, you let mosquitoes feed on it, and before they feed, you have either a control antibody you mix with the blood or these anti-fusion loop antibodies you mix with the blood. They ingest, they do whatever they do. 2 weeks later, you look for the appearance of oocytes. And in the wild type, I think this is with a scrambled peptide, but we've done it with a scrambled peptide. These are how many oocytes you get. With fusion loop, you get reduced, and if you went to higher concentrations, you get even more reduction. And we now have even better numbers for this. I think, well, we've moved on with this, so now we can get blockage at 12.5 micrograms per mil. Now, how would you use, let's see, where am I gonna go? Let me just go back. I just need to explain how this might be useful to stop malaria, right? I didn't do that yet. And I'll be corrected if I'm wrong here, but most kids in places where there's lots of malaria get infected when they're very young. I understand there's a vaccine that's just come out that's maybe going to help in some ways for that. And they get it from a mosquito that bit their parents, probably, that, somebody that they're living with. So that if you could get antibodies against HAP2 in those parents' blood by vaccinating them against HAP2, for example, or against some other proteins that are involved in sexual reproduction in the gut of the mosquito, like maybe against the zygote, you can block transmission. And so that's one of the goals. It's a totally different thing. It's an altruistic. You're not protecting, if you get the vaccine, you're not protecting yourself against getting malaria. But what you are doing, if you could do it in a community, you could suppress the spread. The idea I understand with malaria also, again, I can be corrected here, is that it's not going to take one thing that's going to solve the malaria problem. You got to have multiple ways to try to stop it. And if you don't get it out of the reservoir, it's always going to be tough. Or if you don't stop the reservoir from being able to spread it. So that's the idea for this transmission blocking vaccine strategy. Well, it turns out I had given a talk about this at a Gordon conference about HAP2 several years ago. And this fellow named Felix Ray had talked with me and asked me if I was interested in collaborating, maybe to get the structure. I hadn't met him before. He was with Thomas Cray, his postdoc. And I'd started a collaboration with Timothy Springer, actually one of the founders of Moderna. I didn't know anything about that, who was also trying to get the structure of clammy because it was easiest to do. It turns out you cannot get plasmodium proteins, at least HAP2. You just can't get bacteria or any other kind of cell to make it in a soluble form. It's just impossible. It's been one of the banes of people who work on that strategies. But unbeknownst to me, Felix started working on it. And they found they were able to get it expressed, the ectodomain of clammy HAP2 expressed in cells. And what they found was that the ectodomain, which doesn't have any transmembrane domains, inserts into lipid bilayers. Makes you think maybe that fusion loop. And so they went on then to crystallize it and get the structure. And so here's the structure of the Chlamydomonas HAP2 in trimer form. I'm just showing you one of the subunits. And here's dengue. Here's some leaky forest virus. This is that part that folds up that was down like that. Clammy's got one of those. So-called. This is actually structure that we worked with. When we did it with Felix, we weren't able to get all this structure because they had to use trypsin to do a little, to get crystals for them. They had to break it. They had to hit it in a few places to get crystals. Tim worked out a way to get crystals. And now we can see that it's got these nice big, these are full of hydrophobic residues up here, which if knocked out, block fusion. But it's a class 2 fusion protein, which was very exciting. Turns out you're never alone in science. So two other people had also they used homology modeling to decide that Arabidopsis HAP2 is a gamete fusion protein. And also Jennifer Pinella, who's now a postdoc in my lab, studying tetrahymena, also used homology modeling to realize that the tetrahymena HAP2 was similar to a viral fusion protein. So it was sort of in the wind. So then next question is, which I think is something that's really exciting now. So clammy and probably many of the organisms on Earth use HAP2 to fuse their gametes. But that happens at the surface of the cell. It's not happening in an acidic environment. And you don't want HAP2, like I said, HAP2, it's going to needs to go like this. You don't want it to be triggered any other time except when it's in the right place at the right time. So how are you going to do that? And one way that would make sense is make it so that somehow the adhesive interaction, the species-specific interaction, is where you regulate it, how you regulate it. And we don't have definitive evidence for that yet, but we got some pretty good evidence. First of all, we were able to figure out ways that we could see the HAP2 trimer in clammy after fusion. So this is the monomer. It actually appears as two different molecular weight bands. Only the upper form of the monomer of non-trimer HAP2 is the one that's on the surface. But we could see trimers. Jun Zhang in my lab worked this out. But if you boil them, they go away. But anyway, and not very many made. Only about 8% of HAP2 gets turned into trimers during a normal wild type fusion. So it doesn't take much. What she found was, though, if she mixed fused one defective plus gametes with wild type minus gametes, she didn't get any trimers. We don't get any fusion, of course, but we also don't get any trimers. So that told us that all this adhesion, the activation of the cells to make their mating structures, to get rid of their walls, all the cyclic AMP had nothing to do with trimer formation, although we think maybe it does a little bit, and I won't talk about that. But you actually had to have this mating structure adhesion event go on to get trimers formed. So then that led us to ask, well, what's this protein on the minus gametes that's binding to fuse one on the plus gametes? We did the kind of experiment, Yanji Liu in my lab in Texas, did the kind of experiment that I think at least used to be the standard thing you would do if you wanted to find out two proteins that bound to each other. We biotinylated, let's go back. We biotinylated minus gametes, so only on the surface. So all the proteins on the surface of minus gametes were biotinylated. And we used the fuse 1HA plus gamete, and we mixed them together. Then we solubilized and we pulled down the fuse 1HA, and then we looked with streptavidin to see were there any biotinylated proteins that got pulled down. And of course, there were lots, but there was one that stood out. But of course, it could be anything. It could have, well, it wasn't actin, but then we could go back to our transcriptome, because we got mass spec and we knew this was gene CREO3 G176961. And fuse 1, as you can see, is expressed only in plus gametes, upregulated with activation. HAP2 is expressed only in minus gametes. And this guy was only in minus gametes. So that, oh, maybe we got something. It's non-vegetative cells, non-plus gametes. Maybe this is something that we can really work with. And as you might have guessed, we did. We showed that if we have HAP2 flag gametes, now we rescued fusion. We made a MAR1. Oops, I'm getting ahead of myself. Let me go back. So we call this protein minus adhesion receptor 1, single pass transmembrane protein. Doesn't look like anything else. So it really does look like it's species specific. We bought the mutant for $100 from the Chlamydomonas clip mutant library. Just email, please send us the mutant. We got the mutant, characterized it, and I'll show you some of it in just a second here. But we rescued the mutant with MAR1 flag, and then we mixed them then with fuse 1HA cells. We IP'd with flag, and we pulled down fuse 1HA. So sort of repeated the first experiment with biotinylation. But then we also showed that recombinant proteins made in bacteria, we could do the same thing. We could have a GST tag. One of them was GST tagged, and then we pulled down with GST, and then probed with a HIS, and we saw that we pulled down MAR1. So the proteins interact in vivo with each other in endogenous protein, but also made in bacteria they interact. So there's a direct interaction between fuse 1 and MAR1. And we went on to show, this was done by Yanji Liu and Jennifer Pinello, that the MAR1 mutants are incapable of undergoing mating structure adhesion, and they're incapable of fusion. So it looks like it's the right thing. It's localized on the minus mating structure. Again, these are minus gametes. They've got HAP2. They've got now MAR1 there. And MAR1 is a protein that binds to fuse 1 on the plus cell. So then maybe MAR1 and HAP2 are associated, and indeed they are. So that now we can have minus gametes that have MAR1 flag, HAP2HA. We can do a pull down with MAR1 flag, and we pull down HAP2HA. And you can see that here. And it turns out, as I mentioned, there are two different forms of HAP2, an upper and a lower form. Only this form is on the surface. And that's the one that gets pulled down when you pull down with MAR1 flag. We can't do the reciprocal Jennifer Pinello went crazy trying to do the reciprocal. We don't understand why it doesn't work, but we're convinced that this is a real interaction. And so there's a biochemical interaction between the two. And also in MAR1 mutants, the protein is expressed at lower levels. So we've increased the brightness here so you can see it. And it's mislocalized. It just doesn't get to the right spot. So HAP2 is dependent both functionally for its delivery and trafficking to the right spot, and biochemically anyway, because they do interact. So the model that we have now is that on the plus mating structure, which has fuse1, it interacts with HAP2, with MAR1. Fuse1 interacts with MAR1 in the minus mating structure. MAR1 interacts with HAP2. We like to think that maybe there's some talking going on here to regulate the function of HAP2 so that it can flip out like it's supposed to do during fusion. And the way to think about it is then that maybe evolution has had two ways to play with things. One is it's got this GEX2, at least in plants and unicellular algae, it's got this GEX2 adhesion protein that can then maybe, if you've got another species, I mean, if you've got a changed organism where maybe your gamete of the opposite mating type has a new receptor on it, it might be able to bind to GEX2 because GEX2 also, this is gross speculation, but that's what could be going on there. And Chlamydomonas anyway, the MAR1 is really species specific. I mean, we can't find it. We've looked really hard. We can't find it anyplace else. So maybe during evolution, other things have gone on like that. For example, in mouse, a Zumo on the sperm binds to Juno on the egg, and Juno happens to be a folate receptor. So mice and us grabbed this other protein that was out on the surface to use to bind. And the Zumo, I think, is more conserved than Juno. So it's that idea. So that's the idea that we have. So that sort of to summarize very quickly, Chlamydomonas been able to help us look both below the viruses and up for vertebrates, although I don't know. Chlamydomonas thinks it's pretty good too, I think, in terms of looking up. And maybe come up with some ideas about how evolution might have used these different classes or groups of proteins to bring about sexual reproduction. But the other thing that's neat about this is that these fusion loops, they're important in a bunch of different places that are bad. All these parasitic protozoa, all these guys, I'm pretty sure they're all using it, and all these guys are good. But it's occurred to me there might be a way to, this is a vulnerability, and there might even be a way in my dreams to target drugs that would be species-specific HAP2s, for example, where you could think about blocking. And all these protozoa, Amera causes, I think, a reduction in about 20 or 30% of the economy of the poultry industry. And I think if we could work on that as a transmission blocking vaccine. So I'll just stop there. Lots of folks have been involved with this. UT Southwestern, I mentioned Nick Grishin, Jimin Pei, Zhu Ning was an important guy too in this, and Yanji Liu, I mentioned, was huge in that. We've worked with folks at the Pasteur Institute, Felix Ray and his colleagues there. Juliette Fedry was critical in the crystallization, and then Oliver Bilker, Rita Tarwari, Andrew Blagborough, who we're working with again, continuing on transmission blocking. And then we worked with Oliver Bilker and this group at the Wellcome Trust to do that RNA-seq. And then now in my lab, we have Piyush Ranjan, Mayank Awasthi, who are working more on the cilia section, Jen Pinello, and June Zhang did a lot of the work with the fuse1 and trimers, and she's actually now taking another position. So, and I'll stop there.
The significance of the trimer. Talked about the HAP2 trimer. It seems like an interesting twist to the story. Is it for grabbing onto enough membrane to induce?
Good question. It takes probably a lot has been done with the viral systems. you might guess. And I guess it takes at least three trimers to be able to get a fusion pore formed. Because you have to, if you just stuck up one. And so, and I've tried to understand it too. And actually, when each of the monomers, so you got three of these monomers are going to be doing this. When they're folding up, a part of one monomer interacts with the groove that's formed by another monomer. So they really can't do this on their own. They really do need to get to the stable post-fusion trimer. They need to be able to be in this other conformation. And I'm sure that other people could give you a much better answer than that, but that's my best. Yep.
So on the activation of so one, you're hypothesizing that it's some sort of conformational change that comes about from the interaction between them. But I guess another possibility is that this thing is activated. It's got this fusion loop out there. You need to bring another membrane closer. So it's not the interaction so much as the adhesion.
That's an excellent point.
So can you use like a different adhesion molecules to pull the membrane together?
We've given quite a bit of thought to that. It turns out that a group at the NIH with Leonard Chernamordik and also Benny Poblivowicz, who discovered another class 2 fusion protein in C. elegans that's essential for syncytium formation. In that case, HAP2 or the F1 has to be present on both cell types to get fusion, and their fusion loop is not hydrophobic. But it's still, and they don't yet know if it forms trimers. Presumably it does, that's the model, but they haven't seen any trimer formation. So I think in vitro anyway, with the ectodomains, if you've got the monomer around very long and there's a lipid bilayer around, it's going to form trimers because they're going to presumably stick into it. And so it's possible what you said is what's going on. And one way to test that, I mean, it's a neat thing, is to add liposomes, for example. Because maybe then it just needs something to stick into. It turns out that we think gamete activation might loosen things up a little bit. And so, at least based on the viral systems, you really have to have that thing protected. And also, I guess another thing that argues against that is that with MAR1 minus mutants or FUSE1 plus mutants, the cells adhere, they both make their mating structures, and at least with FUSE1 mutant anyway, they're just banging their things together like crazy. So if there were a HAP2 with its fusion loop out there, it just seems like that interaction would do it. But maybe it's not long enough, maybe it's not stable enough. And so we cannot rule out what you just said. With all the viral systems, they're all, at least with dengue, it forms a trimer where one end is hiding the fusion loop. In other places, there are other proteins that do it. But anyway, yeah.
Is HAP2 and the others always in the membrane, or is there some kind of like I don't know, I'm not familiar with the life cycle or like the start of mating here. So like in Saccharomyces, we have sensing of pheromones and stuff, right? That then we get expression of these fusion related things. Are these guys also controlled in a similar way, or are they kind of always on the membrane just waiting for an interaction to happen?
So I forgot to show my life cycle slide. But anyway, we grow them vegetatively. They don't have any expression of HAP2 or any of these other proteins. They got the cilia, they swim around and all that. If in nature, teleologically, the way we think about it is when the nitrogen source, when their nutrient source goes away, their pond's going to dry up, they're going to die unless they can form a zygote with its thick cell wall. So then if you put in the lab, we put them in nitrogen, ammonium nitrate deficient medium. They all undergo a synchronous 1 more or 2 divisions, and they're all gametes then. they've turned on all these gamete-specific genes with these transcription factors. And one of the genes that gets turned, all these genes get turned on. And so when we look, they're there with tags that we have. They're all there at the little mating structure ready. We don't see them anyplace else in wild type cells. They're on the surface. If we treat with trypsin, all of these proteins go away very quickly. And actually all these proteins go away within about a half hour after fusion also. They get degraded. So
Is the differential expression you see plus and minus, is that driven by the differences in the mating loci, or is there like other things that are driving the only seeing expression of minus and the other one in the plus?
that whole thing hasn't been worked out yet. But anyway, what we do know is that in the minus mating type locus, this mid gene shuts off all the plus gamete specific thing. Whether or not it does it directly or whether it turns on other transcription factors or represses them to make that happen, we don't know. In plus gametes, which don't have mid, this other set of plus gamete specific genes get turned on. We don't know what those are.
Ever tried to do like an experiment where you rescued the effect in the other like by expressing the
It turns out, so can we put fuse 1 and minus gametes? Can we put hap2 and plus gametes? We can do it, but they won't express it.
different promoter or something?
Yep. We almost everything I've shown you today was with the endogenous promoter, but we've also put things under the expression of a heat shock rubisco, which is a pretty promiscuous kind of a promoter, and they just won't get expressed. Well, probably what happens, they get expressed and they get degraded right away. So it says that they probably have some things that other things that are gamete specific that help them get to where they need to go. That's how we're looking at it anyway.
The picture you had is that substrate and R1 interact somehow at the beginning, during the end. Know something about how they might interact based on the structure?
We can get predictions of what MAR1 looks like again from RaptorX, and I haven't got tried to get my brain around them yet, but we don't really. We don't have any idea. We don't yet know if they're both monomeric when they're there, if they're dimers, and if we should be able to figure that out. And actually, I started to bring this up before, but we're lucky that Felix Ray's lab, who's really good at making proteins in vitro, is starting to make MAR1 and hap2. I'm sorry, MAR1 and fuse 1. I can't keep them straight. They're making hap2 and beginning to try to find out which parts are responsible and all that.
You mentioned for at least one of them that there was a really short intracellular domain. But I guess I'm thinking there must be signaling going on. So does intracellular domain tell you anything about?
No, with fuse 1, I don't know. One of the other things about this system that I've realized is that with most of the adhesion proteins, the cell-cell adhesion proteins that we've heard about, that we might be familiar with on vertebrate cells anyway, or a surface ligand and a receptor binds to a ligand binds to its receptor, or two adhesion molecules interact. That triggers a bunch of stuff inside intracellularly to get things to change on the surface or get the properties of that protein to change, maybe. But usually it's through a protein kinase dependent pathway or something. Some kind of signaling event that's going on. With the virus, with hap2, I don't know that you need that. Because this thing, I think, has the energy to do this built into it. Do this folding. And so it could be, if you simply bring fuse 1, you got fuse 1 and MAR1, if you get them together, fuse 1 doesn't really have to do anything except be there. I don't think you need to talk. I don't think fuse 1 has to do anything in the plus cytoplasm. Now, MAR1, we don't know if it's interacting with hap2 in the ectodomain or both. And so one idea is, though, that MAR1 binding to fuse 1 tweaks hap2 in a little way, and then hap2 does its thing, maybe with no signal. It's all just built into this little machine, pre-energized. But that could be wrong.
Right. Thank our speaker one more time. With us.
Thank you for the questions. Yep.
else in the chat?
A little what?
That tweaking that you mentioned is the equivalent of an acidic pH.
Correct.
So
It's
whatever
interesting.
that little tweaking is doing is
It's letting this
essentially
happen.
similar to what a pH change is doing.
Exactly. That's right. I should have said it explicitly, but but we don't, it turns out that it's,
What's about-
lots of people have been studying, of course, how these bad viral proteins work, but it's still not clear which exact residues in yellow fever or dengue or Zika, or I forget the other ones that people study, exactly which residues are responding to the low pH. I'm pretty sure it's histidine, but they don't know which one, and then maybe several of them. And so I don't know if it's even going to be in the same locations.
Sure, sure.
Who's next?
Yep.
Yeah, you just need it.
That's right, exactly. You need a little, that's right,
You
that's right.
need it to get over
Right.
the inhibitory region.
Right.
Whatever's keeping it. straight.
Whatever interactions along all those different residues are keeping it happy. And that way you have to make them unhappy.
Right.
So they're going to go to this other way. Yep. Yep.
Very interesting.
Thank you. Yep. Yep.
Thanks, Bill. That was.
Thank you.
terrific.
Thanks.
Appreciate it. That was.
Yep. Yep.
very-
Thanks. Yep.
as I-
Good questions too.
Yours